Really cool. All right. Hello, everyone, and welcome to the Center for Reproducible Biomedical Modeling's monthly seminar series. In this series, we've been sharing tools that might be useful in your biomedical modeling endeavors. And today we're going to hear from Dr. Jin Xu on an SBML network viewer, and also from Dr. Matthias König on a package called SBMLutils. All right, so just to go over the format for the seminar, for today's session, we'll have two 20-minute talks, and these will be followed by about 15 to 20 minutes of discussion. As always, our goals for this session are to share common challenges that we all have in the biomedical modeling domain, try to share our own experiences and solutions, brainstorm new solutions together, and ultimately build a more collaborative community of modelers. And please hold your questions until after both speakers have shared their presentations. Also, please mute your microphones during these talks. During the discussion, you're very welcome to post questions in the chat or to unmute your microphones and ask questions to speakers directly when there's an open, when there's a lull in conversation. If you have a question or comment, please do share. Someone else probably relates to it and might be able to offer some useful advice or expand on your ideas. All right, now on to our speakers for today. So we'll begin with Dr. Xu, and then we'll hear from Dr. König in the second half of the session. Dr. Xu works as a postdoctoral researcher in the SAR lab at the University of Washington, where she developed software to support biomodeling alongside both experimental and computational collaborators that she works closely with. She has developed a method for generating synthetic networks that resemble biological systems, and is now working to create a tool to visualize network models. Today, she's going to present on this visualization tool for reaction networks, which provides support for SBML models. So welcome, Dr. Xu. You may share.
Thank you, Veronica, for the introduction. The title of my presentation today is Implementing Plugins in a Reaction Layout Tool, Coyote. We have developed a Python-based reaction editor and viewer. Coyote is based on XX Python. You can add, arrange, and modify reactions as you want. So on the canvas, you can put a compartment and put on nodes and connect those nodes with reactions as you want. It can display modifiers. For example, if you click on a node, all the information of the node will be shown on the right-hand form. And then all the information about this node will be shown, like the ID, position, size, and also field color, border color, everything about this node. So you can change those values to modify this node, like position, shapes, and then you can also drag this node to a larger size or move its position with your mouse. So it includes full undo and redo zoom function. You can also import or export models to JSON, SBML, antimony files as you want. And it's cross-platform, so you can use it on Windows, Linux, or Mac. Coyote is extensible and powerful due to its plugin API, where third-party developers can include new functionality. So the plugin system makes Coyote extensible and powerful. We have done some sample plugins which have tested our plugin APIs, and each plugin includes 100 to 600 lines. The sample plugins include, but not limited to, random network, auto layout, export antimony, export or import SBML files, and other plugins like add reaction, align circle, structure analysis plugins. Is it easy to write a plugin? So this package, everything is written in Python. So you can write a Python script to add a plugin to this viewer. And the Python API is straightforward and well documented. You can see more detail online. For example, if you want to add a node, you just use this add node function, include all the information necessary. For the parameters, you can see it's well documented, like the network index, ID of the node, field color, and so on and so forth. So here is an example code that shows how to add a node randomly. So if you want to add a node with a random position, you just do like this. So use add node function, let it know the network index, node ID, size, field color, border color, position, and the positions are randomly assigned. So this function is used to add a node with random position, which is useful for the coming plugin I'm going to introduce to you. So the random network plugin can let you generate a random network on the canvas composed by 4 types of master actions: uni, bi-uni, uni-bi, and bi-bi cases. So if you go to the plugin section, choose the random network plugin, you can design the number of species, number of reactions as you want, also the certain probability for each type of master action. Just make sure the sum of the probabilities of those 4 types of master actions should be equals to 1. And the random seed can also be signed. Like if you want to visualize or analyze exact the same random network, then you should choose the same random seed. Then you can get the exact the same random network. As a visualization tool, we don't want to have those nodes overlap with each other and reactions cross with those nodes. So we also have another plugin. It's auto layout plugin. So with this plugin, it can automatically lay out those network on canvas in a better way. So it has some parameters, so you can adjust the whole size of this network by the scale of layout. And you can also optimize the distance between nodes by the K value. And you can change the maximum number of iterations for each run. So once we have a better view of the network on the canvas, maybe you want to save this network, you can export this network to an antimony file with the export antimony plugin. So if you press show button, this antimony string will be printed on the screen, and the copy button can let you copy the information to a clipboard, and save button can save this network to an antimony file to local in your computer for further analysis. For example, we can import the antimony file to Telegram, then you can analyze it and do further simulation in the software. Because SBML file is more widely used, so we also have developed a plugin called Export SBML plugin. So use this plugin, you can save the network from the canvas to an SBML file. If you press show button, then the SBML string will be printed on the screen, and you can copy it to clipboard and save it to local as well. And those saved SBML file will be available for further analysis. So in order to check whether the generated SBML file makes sense, we have imported to Capacity, a generally used software. So after import this SBML file, you can go to the model section, diagram subsection, and see how it looks like. And the shape, position, color looks exactly as we expected. That means our SBML file generator works. And we also have import SBML plugin. So once you have an SBML file, you can import it to this tool to view it on canvas. So you can just press show button, then the SBML string will be printed on the screen. You can copy it to clipboard and do the visualization button. Then this network will be visualized on canvas from the SBML file you imported. So this is exactly as we expected. So we say it's self-consistency between the export SBML plugin and import SBML plugin. Our tool also support alias node. So what is alias node? Alias nodes are the common nodes among different reactions. For example, the simple example, this network includes two directions. This one is by unit case and another direction is by case. Then they have a common node x2. So if you right click on this node, you can select the split on directions. Then this node will be split to two nodes with the same ID. So this alias node function is kind of helpful for large scale networks. So if one node with many directions, it's kind of hard to visualize it with many directions cross each other. So this alias node function is kind of helpful. Then we want to check if our export and import SBML plugins will work for this alias node function. So we try it again with the export SBML file and then import the generated SBML file to capacity, the shapes, everything as we expected. And we import it to our viewer again, and it's also as we expected. So the import and export SBML plugins works for the alias node function. In our tool, we also support different shapes of nodes. So I have already shown you the default node shape, rectangle. We also support circle, a triangle, hexagon, lines, different shapes for nodes. So this example is a network with hexagon node. So we want to try if the export and import SBML plugins work for this type of node. So again, we export this network to SBML file and import it to capacity. It is exactly show the hexagon node shape, and we import it to SBML plugin again. Then this network is as we expected. So as a short summary for SBML components for our tool, the export or import SBML plugins support SBML files in level 3 with layout and render information. The layout describes the position and size for different graphical objects, including compartments, species, nodes, and directions. And render describes the field, border, color, and line styles. The generated SBML files are compliant with capacity as we have cross-checked, and it also supports alias node function and node shape property. So far I have introduced the SBML compliance for our tool. Next, I want to introduce a little bit more about plugins so that you can see further, like, for example, what kind of things the third party can extend this viewer. So the add direction plugin can help you add a direction on the canvas simpler. For example, if you want to add a uni direction, you just put two nodes on this canvas. Without this plugin, it will take several steps. Like you need to select X0, click the rectangle button, select X1 node, and click product button, and then click create direction button. Then this direction can be added between these two nodes to generate a uni direction. Well, with this plugin, add direction plugin, it can make this simpler. You just need to click uni button, then click node X0, node X1, then a direction will be generated. So add direction plugins allows you to add 4 types of mass directions: uni, by uni by cases, simpler. Okay, so this plugin kind of make this adding direction process simpler. And then for visualization perspective, we'll have a plugin like Align Circle plugin. So if you have a network and canvas, you can select several nodes of it, and then you can align those nodes to a circle, either with a default radius or a desired radius as you want. We also have some plugins to help with analysis of the network on the canvas. For example, the structure analysis plugin. So once you have a network on canvas, you go to structure analysis plugin, you click compute metrics. It can compute stoichiometry matrix and conservation matrix. So stoichiometry matrix can give you the information of each reactions, like for each reaction, which nodes are the reactants, which nodes are the products. And on table 2, it also gives the conservation matrix. It gives the conservative moieties, conserved moieties. For example, for the first conserved moiety, if you select this row, you can choose a certain color, then you can highlight the nodes included. But this circle is from ES to S1 to S2 to ES. And then if you click clear button, then you can unhighlight those nodes. So in summary, I want to have I want to highlight the advantage of our tool. It's Python-based, it's extensible with plugins, can be customized and extensible by the third parties. It has SBML compliance level 3. So because it's based on Python, so you can write all the plugins by Python and use all the packages in Python. So it's kind of helpful and simpler. And for example, the auto layout plugin, it includes network X package for everything to make this writing process simpler. Compared to other software, we have similar viewers in this field, like Kapasi. I have cross-checked with Kapasi because Kapasi is so far the only one has SBML level 3 with layout and render information. So I have cross-checked with Kapasi with the network SBML files I have generated. Well, Kapasi does not have plugin for third party to extend it so far. And Astra and Newt, they are both website based, so they don't have plugins. Astra also do not have SBML support. vCell does not have plugin. Cytoscape, Cell Designer, they have plugin system. Well, they are written in Java, so it's different from our tool. And Password Designer only works for Windows platform. So in the future, we are actually trying to enhance our software Coyote to visualize a large-scale complex reaction network or pathway. So far what we can do is we can design the different shapes of node. We can do the aliens function. We can do the nodes cross with the compartments. Well, does it work for really large reaction networks or pathway? So usually, if the network is very large with many nodes, there will be many reactions for each node. It's kind of hard to visualize it. Maybe the aliens reaction function is helpful so that, and we can do further about this and try more real networks and see how this software would help. So if you are interested in this software, the code is available online from GitHub. You can download it and you can see more information from our well-documented documentation file. And you can pip install it. It's coming soon, so it's easy to install in the future. Okay, thank you so much. We would like to thank all the people who have contributed to this work. First of all, we would like to thank the generous support from NIBIBU24 and our PI, Dr. Herbert Sorrell, and all the people who have contributed to this. Gary, who has developed the software mainly, and I was working on the plugins and SBML compliance. Nan was working on the shapes of node, and Carmen was working on documentation, and Carmen, Evan, and Claire, they were working on the plugins as well. We also would like to thank the generous help from Lucian Smith and Frank Berkman. Thank you so much for your attention. Do you have any questions or comments?
Thanks so much, Jin, for a really excellent talk on Coyote. It looks like there are a lot of opportunities to extend the software. We're going to hold questions for the time being, just until after Matthias's talk. But if anyone does have questions, they're welcome to post in the chat now. Otherwise, we'll get to that shortly. All right. So we would now like to welcome Dr. Matthias König. Dr. König leads a research group for the systems biology and systems medicine of the liver, and previously he's presented for our series on his executable simulation model of the liver. He's also very heavily involved in creating tools to support the use of standards in systems biology modeling, and is an active member of the Combine community leadership. So today we're very glad to have him back to present on a set of Python utilities that facilitate work with SBML models on tasks such as model creation, annotation, or generating easy to understand reports. So welcome, Dr. König.
Thanks a lot for the very kind introduction. Okay, I hope you see the screen, and then yeah. Today I will talk about SBMLutils. These are Python utilities for SBML. I assume most of you know what SBML is, but a quick reminder. It's basically the systems biology markup language and consists of a core language and packages which provide additional functionality, like for instance, composite, the comp package for hierarchical modeling, distrib for distributions uncertainties, layout for layouts, like we just saw, or the FPC package for constraint-based modeling. I would say it's the de facto standard exchange format for computational models in biology, and many tools support it, over 300. There's a large ecosystem of tools, and so I would say this is, if you build computational models or want interoperability with other tools, you probably should work with SBML. Yeah, so despite SBML being like this de facto standard, I found it quite difficult to create SBML models. So like the basic question is like how to create an SBML model, and this was my main question when I wrote my first paper, and I never heard of SBML, and I had a computational model, and the reviewers wanted SBML. So there are different approaches, but all of them didn't work for me, and that's why this SBMLutils package was developed. So there's basically text-based methods like antimony, where you have basically a large text block where you write your equations and define your SBML content. Problems I see with that is often that there is no validation, you don't have type hinting, no auto-completion, and it's currently limited mainly to SBML core information. So there are certain limitations with it. It's very useful tools for small models, I would say, small SBML core models. And I would say, like, if you're new to SBML, this is the go-to standard. Then there are the visual tools, Copasi, the most popular, I would say. Cell Designer also very famous, but unfortunately only supporting SBML level 2. Yeah, these are GUI-based, and a main limitation of GUIs is often that you have no or very difficult programmatic interactions. Yeah, Copasi has an API, but I have to say this is very complicated to interact with that, and I'm not sure if I even would call this an API. But yeah, it is, but it's basically the C code, like it's C code exposed. Okay, and on the other hand, you have libSBML or also JSBML, and these are very low-level libraries, and there are certain problems with that. The one is that you need to write a lot of code. It's basically, like you can see here on the right, just to create a reaction equation with one product and one modifier and so on. You basically have to write 10 to 20 lines of code, and this is like it's just you have to write a lot of code because it's very low level. It's very powerful, but it can do everything SBML can do, but it needs an abstraction layer, in my opinion. And one problem with libSBML especially is that there are many silent failures. And this is a problem in the design of libSBML. And if you expose everything just to Python via Swig interface, this is very unintuitive for Python users. So most operations just silently fail. Like for instance, if you want to write an annotation and you don't have a meta ID, it just fails and you will never know. And so often you have just models where your information is missing and you will never know about it. So these are 3 approaches. For me, none of them really worked because what I wanted was more like something which covers all the packages I can use it programmatically to build my models, and especially it has things like unit support and so on. This was the basic idea why I developed, or the basic reason why I developed SBMLutils, and I would basically call it a collection of utilities for programmatically developing SBML models. It's more like, okay, it's one of this fourth option, but for a certain niche. So it's like if you're a modeler who has programmatic capabilities, then I would say this could be one of the tools to create SBML models. The main features are model creation, manipulation, model merging. It supports basically the SBML core, FBC, comp, distrib, layout, and groups also, like basically most of groups, but this is still in the developed branch. It has especially unit support. It's very strong unit support, so that it's highly encouraged to have units on everything. You have type annotations in the complete code base, and annotation support in the language. So there's basically some abstraction layer which makes it easy to annotate your SBML objects. You can create reports, have certain things like interpolations and some file converters. So this is basically it. How would you create models with SBMLutils? I would say it's this layer around libSBML, which allows, like here, in around 10 lines of code to create a complete model, which would normally take like 50, hundreds of lines. And you can basically have objects which abstract all the setters and getters of libSBML and have additional like internal validation, handle all this missing checks and so on, which makes it very easy to write models. Because it's Python, you can of course programmatically generate this code. And this makes it very powerful because often you have certain rules how to generate your models, and you want to create, for instance, if you want to create me a linear chain like of the length 1 until 1000 and create me all SBML models, like if you do try this with Copasi, okay, have fun with that. If you cell designer similar things, if you with antimony, you could create your text strings for that, but I would say like, okay, it's probably not the nicest thing to do. But here it's very easy to just write a Python loop and generate these models. You have validations and model reports. Importantly, we have direct support for model annotations. So there's basically annotation fields on all the species where you can then just mainly use the identifiers org shortcuts to annotate that. Basically you have object level annotations, meaning every object can be annotated. We're restricting that to the Miriam qualifiers for now. We have something like pattern-based annotations, which I found very, very powerful, where you basically can give regular expressions which match certain properties of your object, and then you can annotate everything with that. Like for instance, like if you have to call all your species as 1, as 2, as 3, then you can just provide the regular expression as number, and then say this should be an SBO term for simple chemical. And so like you don't have to write 20 annotations, you just write 1 annotations, but the pattern is basically matching multiple objects. And I find this very helpful, and especially it enforces also you to use certain patterns. For instance, if you call all your KM values with a KM in there, you just match KM and don't have to annotate all your KMs and Emacs and so on. And we do a validation against identifiers.org resources. This I find also very powerful because like KB and INCHI keys and so on, they have certain regular expression which the identifiers should match. And this information is available, but most people are not validating that these are correct annotations. And this is a very easy way to do it. And I find most of the time I type the wrong annotations in there, and my own validation helps me with that. Okay. Another very helpful thing I find is basically data interpolation. This is a problem I often have in research, that I have an SBML model and then I want to drive this with experimental data. And basically meaning I have like experimental data set over time, and I want to use this as an input, basically as an input formula for my model. For that you can basically do I would say interpolation, convert the interpolation in a piecewise function, and inject the piecewise function basically in the SBML model, and then drive a species or an external input via this data points. We have different interpolations. We could do constant interpolation, linear interpolation, or cubic spline interpolation. So yeah, basically very simple, very simple, but in the end, very useful, especially like if you have a lot of data-driven modeling where you want to have certain inputs clamped to certain experimental data. Yeah, overall, I see SBMLutils in like a larger ecosystem. So what I tried with the tools I develop is more like I always try to only solve a certain problem I have and make these things work together. And I think that would say the thing how it works together is basically SBML as an exchange format. So on the one hand, we have SBMLutils, which is mainly for model development, say model building. On the other hand, we have SBML, which is for model visualization. An SBML sim, which is in the end for the simulation, and we just use SBML as an exchange format between these tools. This is also very nice because this allows me and others just using blocks of that. So there are many people who just use the visualization part, but don't want to use my model building part because they have their own tool or use antimony. So you can just use the visualization without the rest, or the simulation without the rest, and so on. Yeah. And this is also very helpful because often for instance, I create my model with SBMLutils and then I simulate them in Copasi and so on. If it's a small model, I want to just do a parameter scan or whatever. And so like the SBML exchange is what was driving all these different tools somehow. Okay, so what is SBML? It's basically an app for visualizing SBML models. Basically has a few key features. The main things I would say, it has like basically this reaction species view, which we just also saw in Coyote. But it also has like a kinetic view, which I found very important, especially in the context of model building. It also has subgraphs and filtering, which I find very helpful for modeling. Support of annotations, and it works also with large-scale models with thousands of reactions. And last but not least, SBML sim is basically the simulator in the end, which would just take SBML models and allow us to do certain things. It's basically like a wrapper around Roadrunner, which supports certain things I often have to do in my research. So most of these tools were more like I had certain problems and I tried to solve the small problems with a small tool. So, okay. Quickly, the acknowledgement. Basically, I would say nobody funded me for that, and somehow everybody funded me for that. It's more like I got funding for many projects, and part of the project was always you have to build models, and somehow a bit of time went into the tool development. So like somehow nobody gave me specifically money, but I was paid by these people while I was developing these tools. So let's say it like that. Okay, so I want to end with a bit of live demos to just show, okay, how this would work. Yeah, normally you would just have a Python code editor, and then you can just write basically your model objects, and you would then get like things like auto completion, like help, what attributes can you use, and so on. And in the end, you basically can just create the model and, for instance, visualize the model. In this case, I'm creating the simple model, which is, I would say it's a Roadrunner example, the Roadrunner example model S1 to S2 with K1 and J0. So it's it's basically very simple, and you can just create this model, and then you immediately basically can get interactively the visualization in the front end. And yeah, like I said, you not only have the species reaction view, and basically here you get all the information about the metadata, but you have also this kinetic information. And this is often very important for debugging. It's basically, you want to know not only that S1 is going to S2, but you're also interested in the kinetic flow of J0, what parameters are involved. And of course, they also like somehow in this kinetic, basically they are in the MathML graph somehow connected. And you want these connections because often you have like just dangling parameters or empty things and nodes which are not connected to the rest. And these are always signs, okay, your model is something wrong. And so you immediately get a visual feedback, okay, something is not working, or you don't use certain objects. And of course, you can like for the small model, it doesn't make so much sense, but you can very easily just create submodels. For instance, you're just interested in the cell, interested in the next neighbors, and then you want to create a new submodel of that, and then you have a new subgraph and things like that. So you can very easily like just create different views of different subsets and just filter based on SBML types and so on, get your neighbors and get certain subsets you're interested in. And this is often very helpful for debugging in the model building process. Of course, you get a lot of validation rules. In this simple example, we basically don't have any units. Like, so if you would want to do this simple example correct, you have to do it more like a full model. And then this is also like a problem, I would say, in SBML model creation. Basically, this is what people are creating. And this is the same model, and we telling people they should do this here. So it's basically, you can see already, these are three, 4 pages of code, and it's just because we have to do things like, okay, we should describe what it is. We should add creators to that. We should annotate the model units. On the compartments, we should give name, be specific, set annotations, write notes of what we did in the modeling process. If we have data, like we want to encode uncertainties, and so on. And then we see like this one-liner of the compartment we saw before. Basically, this is suddenly like something like we have to create basically this. And this is, of course, it's a challenge, but here it's very easy to extend this from, you can start with a simple model and then you just set the additional attributes while you go along. Yeah, similar, like you don't, all the packages are supported somehow, I would say natively. So it's things like you don't have to do an FBC extension, but you can just set a chemical formula on your species. This is an FBC attribute for the experts here, but you can directly set it. And this often makes sense because you want to have like chemical formulas or charges even on a kinetic model, or you want to do clever things like here. Like for instance, this is something like nobody's doing, but I find it something very clever that you, for instance, can use lower and upper flux bounds on kinetic models. You just set them and define the parameters here locally, and you said like, I expect my flux being between 0 and 1000, and then you can set constraints which could be checked by your integrator. This in the modeling, it could also help to constrain basically your model behavior. So yeah, basically it just shows, okay, in reality, basically every attribute of the supported packages is supported. And this was for me also the important thing, that you can go like from simple models to, okay, I want, no matter what attribute I want to set, I can set it easily directly on the objects. Yeah. Next thing which was important is basically a thing like model composition. Here we saw the simple model, which is just S1 is going to S2 via this J0. And then we would, for instance, like to extend this model. And so we would just write our model extension part, which in this case, like, okay, what we want to extend, we want an additional parameter, and we want an additional reaction. In this case, we want just a reaction which is producing S1 as a second extension. And then we just say, okay, we want the combination of these two models. And basically the order is like defining what is overwritten and so on. And so we can just say, okay, we have our one model and we want to extend it. And then we extended it with basically a J1 reaction. And then we can see we have the same, this model is defined already, and we just extended it with a simple reaction. And this is just in a few lines, you can basically extend things. And because this is all programmatic, here, like you can use loops and so on. Like for instance, if you want to do things like, I want to create a linear chain, again, of these reactions and so on, you just create your species, parameters, reactions in a chain, give the correct identifiers, and then can just easily create, for instance, all your linear chain. Okay, you can see here a nice chain of reactions. Yeah, last but not least, yeah, you can also do similar things with random networks and so on. And I find this very helpful, like basically for benchmarking and so on, just to test test what is going on. And you can, of course, also this works pretty well for very large networks. All the model generation is very fast. I would say the slowest part is the comp model flattening and the unit validation. But overall, you can see the model was created pretty fast. But okay, the layout algorithms take just a bit of time. Yeah, here you also have a lot of layout algorithms and so on, and just a few show graphic details. And also we have created a very large random network and can visualize it and look at the metadata and so on. yeah, last but not least, one more minute. We also have a SBML report. We had a static SBML report. Basically, the idea was to have some human readable report of the whole model. And yeah, this was now funded in the Google Summer of Code in 2021. So we are currently working on a new interactive SBML report. And the main idea is just you can upload an SBML and then get something navigatable and human readable out of that. This is a very early prototype, but you would basically get something like just, okay, these are all my, please ignore the visual look of that. It's just more of a proof of principle that this works. But you would get like more information about all the objects which are in here, and especially like also the linking between the objects. So that, for instance, if you go to the species, you can immediately go, this is in a compartment. This compartment contains these species. This is the information on here. I could look at the SBML and so on. I would resolve the annotations and so on and so on. So and this works also like with comp models and resolve the external model definitions and model definitions, and with distrib and so on. And this is, I would say, like one of the main challenges. Last sentence. Yeah, that none of the tools working with comp and hierarchical model composition, but most of my models are very nice comp models. Like one example here, like for instance, we can here just build a grid of compartments which contain species and transport between them. And then I basically in the next step, I used my minimal model which I had and just coupled it to all the compartments. So basically I created a grid of species, and then I can reuse my cell model, which I created as minimal model, and couple it to all the compartments. And we can see this basically here. I have basically my linear chain of compartments, which is connected by diffusion equations. And in every of the cells, I now packed basically a minimal model of, okay, S1 should go to S2, but the external model, like because the chain is already S5, this just goes to S5 and to the submodel S2. Okay, so sorry for taking so long. Yeah, this is basically it, and how all these different tools work together. Thanks a lot.
Thank you so much, Matthias. You have a very special skill for developing tools that make it a lot easier for modelers to perform pretty complicated tasks like writing and documenting SBML. There's a lot of there's a lot of chat going on between Joe, Herbert, and John. I didn't know if you guys kind of wanted to verbally explain what conversation you were having in the chat.
Well, pick one person to start, otherwise all three of us will start at once.
Go ahead, Herbert. You go first.
Okay, thanks. That was really nice, Matthias. So a couple of things about the Cytoscape. Yeah, we were messing around with the Cytoscape plugin just yesterday, I think, because John suggested we'd try it out. I didn't realize it could do a kinetic, it could do a reaction species one. That I didn't realize that. One question about that, does it export the SBML layout and render extension?
Let's say it exports the coordinates mapped to the meta IDs and IDs. But with this, with this you can easily create. Basically, there is an export of the visual information and of the node positions, and from that you could really easily generate layout. But I haven't. Yeah, it wouldn't be too much work. It doesn't exist, but it's not too much work. Yeah, but this is mainly also, yeah, I need this a lot because I basically just want to lay out my models once and then in this tool and then get the information in the SBML. And yeah, for that, I say like this, somehow it's like this chicken and egg thing, because the problem is my SBML models are changing. And what I basically want, I want the layout information from the last version for all objects which are in my next version, that everything is already layouted. Only the new things should be layouted as good as possible to what wasn't there. And so basically writing the layout in the actual model is not helping me because I need the information for the next model version. So yeah,
but it'd be useful though, but you could split it off from that, and it would just be a specific export import, just so that we could exchange between different tools.
Exactly. Yeah, we had the layout import, but I could make a layout export for that.
Yeah, I think it'd be really nice. Yeah. Okay, thanks.
The other discussion that was in the chat was to do with, a few of us have found this similar need with an abstraction layer on top of the libSBML for people who programmatically are developing models. And so Herbert mentions a simple SBML, a package that is PyPy. I have something also named simple SBML. If I haven't put it on PyPy, I was trying to figure out how is it something that could be generally used. But I think there might be some value in having a discussion about what a more unified set of features might be, at least a base set of features that we all build on. Anyway, because I could see us, many folks going different directions with that abstraction layer. So just a thought.
Definitely, and I would say often this abstraction layer is driven by your actual modeling needs, like PySB and so on. They also have like their own language, but it's more like they want their multi things in there. So I often would say like the complexity of your abstraction layer is driven by your models. I can say for me, what was driven is basically the model composition, because we're building a lot of physiological based models, and we just have as sub-models the tissues and so on. And so we want to develop our tissue models or organ models and then couple them to the complete model, and all like in a same framework. And so like for just developing models, there's a lot of things, but for this model coupling and developing submodels, there was not a tool. And the units, I have to say, this is people ignore units, but for me it was more like I can see most of the time I'm just doing the wrong things, and it's especially on the kinetic equations. It's they are always wrong, and also your constraints are wrong, and your events are wrong, and then so on. Because it's most of the time it doesn't really matter. But often it's your model gets much better if you include the units.
It's quite verbose, though. I thought your code was quite verbose, just to do one reaction. So whatever the abstraction layer is, I think if we did one, there need to be multiple ways into it. So simple SBML, you could do that in one line, for example, what you did. And if you don't want that much detail, on the other hand, if you want a lot more detail, then you need to have a bit more of a, you have to let the user add that detail. So this has to be some compromise between people who just want to have a quickly build a model and those who want to build much more detailed ones. So that's the only issue to have to figure out.
Exactly, I agree. For me, it's more like I have a lot of things I do programmatically. Like for instance, like my annotations are just stored in an external database, and I just map them and things like that. For instance, I'm never writing the annotations, but in the end, I'm just going over my IDs and name combinations and look in the annotation database. Do I have something for that? Because for instance, in all the physiological based models, I have a liver, so I don't want to annotate always liver, so I look it just up. If I use liver ID, then the annotations exist somewhere, and I can write them programmatically afterwards easily. So, but I have to. It's not as low level as libSBML. It's an intermediate level.
No, I see
a lot of issues.
I see us using SBMLutils. Jan is in fact here on the call. I think she's building a large metabolic model. I think we would probably leverage SBMLutils for this purpose. So
I had a question about units. So I noticed that, and we exchanged email about this as well, Matthias. I should first say great talk and good work as always, of course. But you have units on species, which seems wrong to me, because in general, for any kind of process, and you'd have to leave biochemical reactions for this, but you can have a species or an entity like a fluid, and you could measure different things. And so obviously the different measurements would have different units. You could measure pressure and temperature and volume. For biochemical reactions, I guess you're always looking at chemical concentrations or molar concentration, right? So, but still, why would you attach it to the species rather than to the property of that species? But I guess there's no distinction between the two.
Basically, the species has I'm not sure I understood this
So
completely, but the species is representing basically something, and this something has a unit. It can either be a
right.
concentration or amount, or it could be something totally different. Some
But it
entity
can't
and
ever
so on.
be two things.
I just opened an issue about this, and like this was a design flaw, I would say, in SBML, but this was before my time. So it's not my fault. But it's more like let's say like the basic rules for SBML that all kinetic laws have to have the same units, and this is basically extent unit per time unit, and this in the end means that all your species must have extent unit. Okay, and people realized, okay, probably this is not a good idea, because sometimes I want to have proteins which are dimensionless, or in items and concentrations and millimolar at the same time in the model.
Right.
So what they introduced is basically conversion factors on species which allow to convert your units to the actual units. So your whole mathematics is always an extent per time, but you indicate on the species what the actual unit of the thing is via a conversion factor. So yeah, I basically stumbled over that. Like there's a post yesterday in SBML development about that, but it's yeah, exactly. I totally disagree, and this is completely unintuitive. But in the end, you just have to look at the unit of the conversion factor times the unit or extent unit to get your actual unit of what you have, and then you can encode basically any unit on species.
Yeah, I mean, this is a very weird way from my perspective, of course, from a composite annotation perspective. We talk about physical entities versus physical properties. It's two completely different things. The physical entity, glucose, is a thing, and then you measure information about that thing, like the chemical concentration in a particular compartment. And that is a property. And so it's the properties that get the units, not the things. That's the way I think about it.
Exactly, but that's
but that doesn't get translated.
It's a
SBML.
design issue in SBML that basically you look at the species as a whole complete thing which has units and means something, and in reality this should be more fine-grained. I agree.
Yeah. Okay, thanks.
But sorry, last sentence about this comes like the people wanted a way to generate ODE equations. It was my feeling when I look back at that, and so it was the easiest and nicest way to create consistent ODEs between tools, and this makes sense somehow. And then in the end you say like we cover all the cases, and if something is different, then okay, in this 1% of cases, then do a conversion factor. So
right. Thanks.
I guess I have a question for Matthias. I was wondering if what you've heard from users about which parts of your software has been, I guess, most commonly used, or the gap that they find filled best by your software.
I'm not sure. Like I would say the visualization is used by a lot of people, basically because it goes up to genome scale models. But Azure was pretty popular, but the problem is for very large models, you have to generate your own layouts, which is very, very cumbersome. So I would say that there's a lot of FBC people using the visualization. With SBMLutils, it's more, I'm not aware of many people using it. Some people use it for model merging. Yeah, but we basically use it for model creation in my group. And I would say that my bachelor students and so on, it's normally you explain them the concept in 2 or 3 hours, do an example model, and then it's pretty intuitive how to do things. But of course, you need I would say bioinformatics, a bit of programming background. Like antimony is something you could write like without having any idea about programming. Here you have to know a bit of Python to do the things.
When you're talking about Azure there, and how layout is difficult, and that's a general problem. It'd be nice if we could all settle on a common layout format, and well, we have one in SBML. If everybody could use that, then we could have pre-laid out. We could have a database of pre-laid out networks as templates that you could then populate for a given organism or just for a given organism, which would be nice.
Yeah. This is basically the problems are the annotations. to tell to John, there's basically the map for Recon 3D for the basically everything in Mammalia is covered by that. There are also the very large E. coli maps, which
Yeah.
are laid out in Azure. But the problem is if you now have a different organism or a different tissue and so on, you have to do an ID mapping. And basically you have to do an ID mapping on the Azure or here the big database IDs.
Yeah.
And if you don't have annotations, which you could use to map the IDs, then it's basically
But at least the layouts could be there, and it would just act as a template on which you could then annotate. It would save some time at least. I mean, that's half the problem is that you can't get
I think the same. Like every time I lay out the glycolysis in the TCA cycle, I'm like, oh man, this is the 30th time I'm doing that.
Exactly. I mean, it can take hours. So it'd be nice if we had some like that.
I mean, don't you want automatic layouts? I mean, we've been doing automatic pathway layouts for decades, really.
Yeah, but we want the ones that look like, yeah, you're right, you have done. A lot of people do this as well, but it'd be nice. I mean, yours look a little bit more like what they are in textbooks, which is what I'm thinking about. But if you could export, so there's this extension to SBML, which probably a lot of people don't know about. I mean, if you export SBML, right, I think.
Yeah, we do, yeah.
If you could add in the future, if you could add the layout and render extensions, then we could load those into other visualization tools then, and then you could actually re-import the layout information to say somebody made a neater version.
I think the problem with auto layouts is that everybody wants to tweak them again and again and again. And so
yeah,
if somebody custom tweaked, here's what the TCA cycle should look like, that would be different than an auto layout. And everybody would like that better, and they would like to save it. Here's Matthias's version of the TCA picture.
Yeah.
And then we could share that rather than the auto layout version, which isn't quite the same.
Yeah. I mean, the auto layout will take you some way, and then you'll think aesthetically, you'll look at it, think, yeah, it's not quite right. Let me adjust it a little bit here and there. But the auto layout would be a good starting point, definitely.
Yeah, I think normally for this auto layout to work well, you need additional constraints on the pathway level. Let's say like that. Basically, you have to use the pathway information to lay out the things. For instance, you know that's the pathway TCA cycle and so on. And often this is annotated like in the subsystems or in the groups of the FPC models. But yeah, you need this basically as constraints for your layout algorithms. And most layout algorithms just work on the node and edge basis, and this is too small. You need basically, or at least the compartments have to be some constraints, and then the pathways in the compartments. And if you lay out these nice, then you need some additional last minute optimization to move the nodes a bit. And I think this hierarchical thing would probably work much better.
Yeah. Okay.
Yeah, but comment to that. What I'm doing in the cytoscape is basically I'm just cutting out subnetworks, and this works for me. Like if it's too big, I'm just selecting based on the pathway and cutting out the subsystem. But I'm not creating the layouts for that. It's more like, okay, to understand it, and basically if you then do a force-directed layout on 20 nodes, it's understandable. If you do it on the complete network, you get just a hairball, and that's not very useful.
So I didn't realize you could cut pieces out. So when you cut a piece out, can you export that piece as SBML?
The problem, the cutting out.
Is that a no?
Because I have certain for the species reaction view, it could work, but for the other views, probably when you're cutting, you make an invalid SBML. So probably I could write it, but it's
right, okay.
Yeah, I could imagine. Yeah, the things are just becoming invalid. For instance, even if you cut out a modifier species, you still have it in the math equation, and your model is just invalid suddenly.
Yeah. Okay. Hi, Peter. Welcome to this talk. Thanks for attending.
Thanks, Owen. Yeah.
Yeah.
It's been quiet in the background here.
Yeah.
We are just out of time, but thank you, Dr. Xu and Dr. König, both for sharing your tools and generating really good discussion. We hope our attendees feel a little more comfortable using them, and otherwise, there are obviously lots of ways to reach out to both Jin and Matthias, I think, to get more information about the tools. We'll reconvene the seminar series in September for a new series that will be focused more on new updates in modeling research. So keep a lookout for an announcement about that coming up in the next month. Otherwise, thank you all for attending.
Thanks a lot. Bye, everyone.